I'd like to invite Dr. Veronica Porreca from Sapienza, University Rome, to talk to us about deciphering the impact of THBS1 THBS2 and PEDF in intrahepatic cholangiocarcinoma malignancy. And this is AMMF-funded research. So, Veronica, thank you very much for coming.
Thank you. Good morning, everyone. And before I start, I would to thank the AMM Foundation to invite me and also to show you our upgrade research work. Basically, currently the molecular basis about the hypovascularity of intrahepatic cholangiocarcinoma is still unknown, but it can be assumed that some soluble factors, including in the extracellular fluid, could be involved in a different regulation of angiogenesis and lymphoangiogenesis in the frame of ICCA. For this reason, to gain science, a few years ago, our lab started to perform the proteomic landscape on tumor extracellular fluid from hepatic surgical resection from ICCA patients. And proteomic data integrated with bioinformatic analysis reveals an upregulation of 91 proteins, in particular proteins involved in negative regulation of angiogenesis. Our attention were focusing on two matricellular glycoproteins, the thrombospondin 1 and thrombospondin 2, that they have anti-angiogenic properties and they are involved in cell-to-cell and cell-to-matrix interaction. But also, we wanted to focalize our attention on the PEDF, the pigment epithelium-derived factor, that is a neurotrophic protein and is considered a potent inhibitor of angiogenesis. First of all, we started an immunolocalization study just to understand in which kind of cells of the ICCA tumor microenvironment could be expressed these three proteins. And immunofluorescence analysis reveals that the overexpression of these two thrombospondines and PEDF are significantly overexpressed in ICCA tumor microenvironment, particularly by cancer-associated fibroblasts. Furthermore, molecular analysis reveals that in the ICCA extracellular fluid, the overexpression of these three proteins, it could be considered a real hallmark of this cancer compared to the extracellular fluid from non-cancer tissue and also from the perilayer cholangiocarcinoma and hepatocarcinoma. Subsequently, we wanted to focus it on the potential anti-angio inhibitory action by these three proteins, and for this reason we performed an endothelium tube formation in vitro. So basically endothelial cells were treated with the extracellular fluid from non-cancer tissue compared with the ICCA1, and we found that the extracellular fluid from ICCA could inhibits actually the epithelial tube formation in vitro. But the question is which kind of soluble factors, including in a tumor extracellular fluid, could be doing that? So we performed a second experiment, this time pre-incubated the tumor ICF with antibody antagonist antibodies direct to the two thrombospondine and the PEDF. The combinant targeting of the two thrombospondine and PEDF by these antagonist antibodies restore the normal vascular network, suggesting that maybe these soluble mediators could regulate the formation of blood and lymph vessels. To confirm this, we set up two different xenograft models. The first one to evaluate the indirect role. So basically, mice were subjected with matrigel plug containing human endothelial cells, plus the bone stromal marrow cells, and also with the extracellular fluid from non-cancer tissue, the ICCA ICF, and the ICCA ICF pre-incubated with the antagonist antibodies. As you can see from the histological analysis, we found that the rate of lymph vessels was significantly reduced after the treatment with the three antibodies. Similar results we obtained also in another endothelial tube formation in vitro, where we found that endothelial cells culturing on matrigel and treated in the same condition of mouse models, we found a number of cells positive for podopline significantly reduced after the treatment with the 3 antibodies. So, to sum up, we could say that the 2-thrombospondine and PEDF expression inhibits angiogenesis and promotes in vivo lymphoangiogenesis by transdifferentiation of blood vascular endothelial cells to a lymphonotic-like phenotype. Furthermore, we performed another xenograft model just to evaluate the direct role. Basically, mice were treated with an ICCA human established cell line, the CCLP1, in presence with the three different kinds of extracellular fluid. As you can see from the pictures, we found that the combinant targeting of the 2-thrombospondine and PEDF reduced tumor growth. But more interesting, we found that we have also a reducing about the neolymphoangiogenesis and the lymph node infiltration. So this study reveals that some soluble mediators contained in the extracellular fluid of ICCA could be involved in the tumor lymphoangiogenesis tumor-associated and also to hinder the vascular network in the frame of ICCA. But the question arises, what are the specific roles of the 2-thrombospondine and PEDF in priming cellular processes involved in the malignancy of ICCA cells? So we started our study focused on the 2-thrombospondine, that they are a complex structure for a variety of ligands. They are matricellular glycoproteins, and these multifunctional proteins are known to inhibit angiogenesis by affecting the cell motility, apoptosis, and the proliferation of vascular endothelial cells. In the context of the cancer, the effects of the 2-thrombospondine, in particular on tumor progression, are multifaceted and sometimes opposite depending on the molecular and cellular composition of the microenvironment. So basically, we started to focus on the direct role of these two matricellular glycoproteins in the cellular function of ICCA cells, I mean in their malignant behavior in terms of proliferation, adhesion, migration, and invasion. We started to analyze the ICCA cell line, the CCLP1, and we treated these cells with human recombinant thrombospondine 1 and 2. And we assay, firstly, the proliferation rate. Basically, after the colony formation assay, we have found that after the treatment with both human recombinant thrombospondine, the number of colonies was higher than the control. But more interesting, we found also that the thrombospondine 2 already at the dose of 100 nanogram on milliliters, the number of colonies was higher than the control. Afterwards, we wanted to evaluate the time course of the cells, and we performed an MTT viability assay. And we found that the time course of the cells after the treatment with the 2-thrombospondine was higher than treated cells. And this data was also confirmed by the evaluation of expression levels of the marker PSNA, that is, was upregulated after the treatment. Then many studies reveals that the 2-thrombospondines are involved in adhesion of numerous types of cells. So for this reason, we wanted to evaluate also the adhesion property of the cells. Basically, we performed an adhesion assay on matrigel thin coating, and after the treatment, we found that after the treatment with the human recombinant thrombospondine 1, we found a higher adhesive properties of the cells, in particular in a dose-dependent manner, and also we found an up regulation of the activity to the cells to adhere on the matrigel thin coating after the treatment with the thrombospondine 2. Then we assay also the migration and invasion properties. So for the migration, we performed an Hund-Helling assay, and we found that after the treatment with the 2 matricellular glycoproteins, the capacity to the cells to migrate was upregulated. But in particular, after 6 hours from the treatment at the dose of 1,000 nanogram per milliliters, the capacity of the cells to migrate was already upregulated. So for this reason, we wanted to chose this dose to perform an invasion transwell assay. And we found that after the treatment, the invasion capacity of the cells to invade the extracellular matrix was upregulated, in particular by the thrombospondin 2. Basically, in this study, we were focused on two different iCCA cell lines. The first one that I show you is the CCLP1 cell line, and then we consider also the HUCCT1 cell line. Basically, these two iCCA cells different in terms of phenotype, because the CCLP1 are principal mesenchymal, then the HUCCT1 that they are epithelial. Then we wanted to evaluate the expression levels of the two matricellular glycoproteins in these two iCCA cell lines. And we found that in HUCCT1, we are an upregulation of the two matricellular glycoproteins from whole cell extract, but furthermore, these cells can secrete the two matricellular glycoprotein in extra space cellular. So for this reason, we wanted to evaluate these second iCCA cell lines in terms of proliferation, adhesion, migration, and invasion. And we found that after the treatment with the two human recombinant thrombospondin, we found an upregulation of these cellular processes, in particular by the thrombospondin 2, suggesting a potential and higher role by this, the human recombinant thrombospondin 2 than the one. Then we wanted to evaluate if the two matricellular glycoproteins could commit HUCCT1 cells forward the mesenchymal phenotype. So for this reason, we consider a marker of the epithelial mesenchymal transition. So we considered bimentin, and we found that after the treatment, the expression levels of this marker was upregulated. But furthermore, we considered also a receptor, IGF receptor, that is involved in oncogenic processes and also in tumor progression by the two thrombospondin. And we found an upregulation significantly after the treatment with the two matricellular glycoproteins. So in this first part of the study, we can conclude that the two matricellular glycoproteins promote the malignant behavior of the iCCA cells independent by their phenotype. Also that the thrombospondin 1 and 2 committed epithelial iCCA cells toward a mesenchymal phenotype, but also increased the expression of the oncogene IGF receptor. Furthermore, that the thrombospondin 2 human recombinant is a stronger enhancer of the malignant behavior of iCCA cells compared to the one. Obviously, we are ongoing in a different field experiments, so basically we wanted to identify at the moment the thrombospondin 1 and 2 cognate receptor and the signaling pathways driving the thrombospondin 1 and 2 induced iCCA malignant cell behaviors. For this reason, we wanted to evaluate this higher activity by the thrombospondin 2, and we considered the cell line HUCCT1. In particular, these two matricellular glycoproteins present binding to domains for the integrins, in particular integrins beta 3 and beta 1, that are involved in cell adhesion. So first of all, we started to evaluate the expression levels of these two integrins from total tissue extra from non-cancer tissue compared with iCCA samples. And basically, we found a significantly reduction of the integrin beta 3, but non changing about the integrin beta 1. This data was also confirmed in our in vitro system, but we hypothesized that maybe the thrombospondin 2 not could regulate the expression levels of integrin beta 1, but maybe its activity. So for this reason, we performed an adhesion assay, this time treating the cells with the thrombospondin 2 in presence of antagonist antibodies direct to the integrin beta 1. And we found a significantly reduction of the adhesion capacity of the cells. The third actor of the story is a PEDF and is anti-cancer effector. He is a ligand for C-viral receptor and play a crucial role in inhibiting cancer cell proliferation and migration. Like for the two matricellular glycoproteins, PEDF was to understand the direct role of this neurotrophic protein in intracellular function of iCCA cells. We found that after the treatment with the human recombinant PIDF, we don't have any effect in cellular proliferation in HUCCT1 cell line. But we found a reduction about the adhesion capacity of the cells, really significantly than the control, and also a reduction about their migration capacity, suggesting a potential oncosuppressor role by the neurotrophic protein. Concomitantly with the assay, in vitro assay, we set up other two xenograft models, this time inoculating mice with the two ICCA cell lines, so the CCLP1, HUCCT1, in presence of the human recombinant PIDF. Actually, these two xenograft models are ongoing, but we're starting to analyze, to characterize the first one with the CCLP1, and as expected by our in vitro assay, we don't found changing in terms of the tumor weight, but very interesting, we found a reduction about the weight of the lymph node. Obviously, this data is going to be confirmed by immunohistochemistry analysis of ICCA cells spreading in local lymph nodes. So about PIDF, we can conclude that PIDF does not affect ICCA cell proliferation and reduce cell adhesion and migration of ICCA cells. And the ongoing experiment is going to be to extend the role of PIDF in the ICCA cells spreading by CCLP1 and HUCCT1 xenograft models, but also to identify the PIDF cognate receptor and the related signaling pathways in ICCA cells, and also to define upstream epigenetic regulators controlling post-transcriptional PIDF signaling. I would like to thank you, my principal investigator, Carmine Mancona, that he gave me the possibility to be here and to show you our results. And in particular, I would like to thank La Dottoressa Eleonora Corbella. She's just a student, but she made a lot of experiments in this project. And also, I would like to thank our team collaboration from Sapienza and also our friends from Università di Catania, Professor Marco Ragusa and his staff, to give us great advice and supporting. Thank you for your attention.